Okay, so the second talk, it's not really the second, but the second part of my talk will be devoted how we take the knowledge that we have accumulated over the last 20 years, understanding the crosstalk between the brain and immunity, into a therapy to Alzheimer's disease, which is based to immune-based therapy or to age-related dementia. So we have a tendency to forget students, so I would like to emphasize this is a real effort of 20 years, numerous graduate students, many from different countries. These are the current team, these are previous team. Many of them are now independent professors in different universities. And our collaborators, some of the work that I will present today is with really tight collaboration with Professor Ido Amit from the Weizmann Institute and Tony Weiss-Curry from Stanford, and also Eran Elinav, the previous speaker, but I'm not going to show data because we are still cooking it. So we know that in all neurodegenerative conditions, all mental diseases such as depression, post-traumatic stress disorder, or neurodegenerative conditions like Alzheimer's, Parkinson, ALS, you name it, there is not a single neurodegenerative disease in which there is not local inflammation. So in many of them, in all of them actually, there is local inflammation. There is a big debate whether the local inflammation is the primary cause of the disease or a secondary cause of disease. Assuming that it's not the primary cause of the disease, so there are numerous risk factors, environmental factors, genetic factors that can cause depression, can cause post-traumatic stress disorder, they can cause Alzheimer's, ALS. So no matter what is if it's the primary or not primary risk factor, we always have risk factors that activate a cascade of events in the brain, which involve the resident immune cells of the brain, and we will get into it, and these are microglia. In all cases, the initial response to the risk factor is inflammation, which is beneficial. And if Eran in the previous talk mentioned Mechnikov was really the godfather of modern inflammation. He was the one who introduced the idea that inflammation is a physiological response, and it's beneficial and it's needed. Only when it gets out of control, it's pathological inflammation. We tend to say inflammation and we equal it with bad response. It's a physiological response. So in all of these cases, there is an initial inflammatory response. However, inflammatory response has a strong tendency, which if it's not resolved in time, it will get into chronic inflammation, which add a risk factor to the overall progression of the disease. So according to our current view, no matter whether inflammation in Alzheimer's, in ALS, in all of these conditions, is a primary or secondary, eventually it is a factor that contributes to disease escalation. Based on the old dogma, whenever inflammation was identified in any brain condition, the treatment of choice immediately was anti-inflammatory drug, whether it's steroidal and non-steroidal. And in all cases, it failed. And it left the community with a big question. If there is an inflammation and anti-inflammatory drug falls short, what's the reason? What have we missed? So these are just few examples of failure of anti-inflammatory drug following a big clinical trial, mainly in the United States. The last one was 2008. All of them have fallen short. So our view, and this is a snapshot of how we view it, and I'll go through it step by step, what we have gathered and other people gathered. Our view is indeed in the brain there is inflammation. However, based on what we know from immunology of wound healing or any other type of immune response, healing of inflammatory response is an active process. Giving anti-inflammatory drug is not the way to resolve inflammation. It's an active process which involves inflammation-resolving cells. So if you want to bring inflammation-resolving cells to the brain, you have to pass through barrier. And what I will show you that there is a unique interface between the brain and the circulation, which is not the blood-brain barrier, but the blood-cerebrospinal fluid barrier, which is a gateway for immune cells. And to activate the gateway, to bring anti-inflammatory cells or inflammation-resolving cells to the brain, you need active immune response rather than suppressive immune response. So according to our view, treating inflammation in the brain with anti-inflammatory drug is like treating a frostbite with ice. So it's counterproductive. So this is summarized 20 years of work that we have done, and let's start with my real talk. So we know that the brain is the bigger consumer of oxygen. The weight of the brain is less than 2% of our body weight. Nevertheless, the brain consumes more than 20% of the oxygen that the body consumes. And in spite of the fact that it's heavily loaded with blood vessels, every blood vessels within the brain parenchyma is covered by blood-brain barrier. As a result of this understanding of the blood-brain barrier, it has been believed for many, many years that immune cells are patrolling all bodily tissue all the time, but the brain is excluded from this patrolling. We know that immune cells patrol the body for two main reasons. One is to eradicate bacteria or viruses before they get into the tissue, and second, to recognize cells that may develop into a cancerous cells. So immune surveillance is a mechanism that protects the body, and their belief was that it protects the body against pathogen and against cells that may develop into cancerous cells. The belief was that immune cells are excluded from the brain because of the barriers. Now, because only a few of you are neuroscientists, the lecture is not just to neuroscientists, so I'll explain every single phenomena. So we all know, even non-neuroscientists, that between the brain and the circulation there is a blood-brain barrier. And when we think about developing drugs, we said, how should the drug pass the blood-brain barrier? However, between the brain and the circulation, there is a very unique and interesting barrier, which is called the blood-cerebrospinal fluid barrier. So basically, this barrier exists in the four ventricles of the brain, at the roof of each ventricle, and its unique structure is in the sense that the blood vessels, the endothelial cells, are fenestrated. Unlike the blood-brain barrier, where they are tightly connected, they are fenestrated. And the barrier is formed by epithelial cells, tightly connected epithelial cells. They are facing the CSF, and this is facing the brain. So it's not endothelial barrier, but epithelial barrier. It was believed that the sole role of this barrier is the formation of the cerebrospinal fluid, the fluid in which the brain is floating, and the fluid that contains all the nutrients that the brain needs. Or there are some cargo protein that bring in and out protein from the brain and outside. So what we discovered that this interface is the place where there is a crosstalk between the brain and the immune system. And we'll get into it later on. But because this barrier was completely ignored as a barrier for immune communication, the belief was because of the blood-brain barrier, no immune cells are allowed to enter into the brain under any circumstances. And as a result of it, when there was an inflammation discovered in Alzheimer's, in Parkinson's, in depression, the idea was, wow, it's a reflection of entry of immune cells to the brain, we should suppress it. Now, when we enter into the field, there is one more issue which is very important. At that time, it was known that in the brain there are myeloid cells. Myeloid cells are like the monocytes in the blood, so they are primitive immune cells. And everyone thought, since we have innate immunity of the brain, it's very easy to live very nicely in the brain without any entry of immune cells from the circulation. So all in all, the dogma was immune cells are not, circulating immune cells are not needed in the brain. They can only be the enemy of the brain. There are myeloid cells in the brain that do the job, and the barrier protects the brain from immune cells. This is the structure. This is the difference between the choroid plexus epithelium, which is the blood CSF barrier, which I just showed you. And we have the tightly connected endothelial cells in the blood-brain barrier, and this is regular capillary. So before I enter, we know already from our work and other people's work that indeed inside the brain parenchyma, in healthy brain, you don't see T cell, you don't see any type of circulating immune cells, only myeloid cells. But you do see in the, and I'll show you that in the blood cerebrospinal fluid barrier and in the meninges, there are immune cells. With respect to the resident myeloid cells, as I said, the dogma was that we have microglia, and if immune cells coming from the brain, they are like the microglia. So my team, and I'll show data, were the first to show that microglia and myeloid cells that come from the brain are not redundant cells. They are not part of the turnover of the microglia. Microglia has their own turnover, which is very slow. And in healthy brain, the monocytes from the brain don't replace the microglia. You see them only under pathology, and they are never becoming macrophages. You have microglia and monocytes from the circulation that become macrophages. Do you need any explanation about monocytes? I will be happy to explain. So all the hematopoietic immune cells are born in the bone marrow. The bone marrow give rise to several immune cells. The most known and the most important for our discussion today, from the bone marrow, we have the adaptive immune system and the innate immune system. The adaptive immune system, you have a T cell, you have B cell. We will discuss today only the T cell. And in the T cell, we have effector T cell, like the CD4 and CD8. The CD8 are the ones that are killing the tumor. They are supposed to kill the tumor. And the CD4 are antigen-presenting cells, but we'll get into it. I don't want to overload you with immunology because this is not the essence of my talk. But just to familiarize you that the immune cells are born in the bone marrow, some of them during early in life, and they mature in the different lymphoid organs. But overall, we will focus today on the T lymphocytes. The immune system has a very important regulation. Because we all have memory T cell and we don't want them to work all the time, so they are under tight regulation, and we'll get into it. Some of the regulation are by regulatory pathway and some by suppressor cells. The other population of immune cells that are born in the bone marrow are monocytes. Monocytes are the garbage, they become mature macrophages, are basically the garbage disposal of our body. They are meant to eat and to kill pathogen, but they have a critical role in wound healing. They change their phenotype based on the environment in which that they are encountering. In the blood, they are called monocytes. Once they mature and get into tissue, they are called macrophages. In the brain, we have microglia. They are myeloid cells. Up until 6 years ago, it was believed that microglia originate from the bone marrow like macrophages. We now know, not from our work, from several other work from 2009, that their origin is from the yolk sac. So the monocytes in the blood are born in the bone marrow, whereas the myeloid cells in the brain, the microglia, comes from the yolk sac. So it's a very early stage during development. So one more issue that I need to explain before we are getting into the brain is I mentioned the T cell. So the T cells are born in the bone marrow. They mature in a very simple organ and very small organ in the thymus. Now, in the 50s, Burnett got a Nobel Prize for suggesting a mechanism whereby the body gets rid of cells that may later on develop into an autoimmune cells that attack the tissue and become autoimmune disease. We now know that he was right, but partially right, because we all have, he was right in the mechanism, but we are not eliminating all immune cells that recognize our tissue. We all have in our body cells that recognize ourselves that are called autoimmune cells. Autoimmune cells are not equal to autoimmune disease. We rarely develop autoimmune disease, but we do have autoimmune cells. So in the thymus, the cells, the T cells, are undergoing a selection. Those that don't recognize any self are dying. Those recognizing with high affinity are killed by negative selection. And we are left with cells in between. Some of them are recognizing self-tissue. This is important because we will get into it. So before we enter into the neuroscience, I just want to summarize. The brain is a tissue behind barrier. There are not one type of barrier. There are the blood-brain barrier. There is also leptomeningeal barrier, but we'll not get it. There are the two extreme barriers are the blood-brain barrier, which is endothelial barrier, and the blood-CSF barrier, which is epithelial barrier. I'll show you that the epithelial barrier is permissive for crosstalk between the brain and the immune system. The other issue, which is very important, we have in the brain myeloid cells, the primitive innate immune cells that are recognizing pathogen, help in healing of tissue. Because of these myeloid cells and the blood-brain barrier, the belief was that no immune cells from the circulation are allowed to enter into the CNS under any circumstances. The other issue which is important for our discussion today, we all have T cells that recognize self, but they should be of low affinity. High affinity may develop autoimmune disease depending on the phenotype, but this is beyond the scope of our talk today. So, in 1950, the belief was, and this is just to give you an history of the field, in 1950, we all knew that no autoimmune cells are allowed to be in our body. If they are, they escape from deletion in the thymus. And it was highly believed that no immune cells are allowed to get into the brain under any circumstances. Paul Ehrlich was the one that first suggested it. We are now here. We know that immune cells are allowed to enter into the CNS and autoimmune cells are allowed to be in our body. So what I'm going to show you. So in the first, I'll give you more a general part. So it's more general and less general than what I've said so far. And the second one, I'll show you how we harness the immune system to fight against brain aging and Alzheimer's disease. Any question up until this stage? Because I have a tendency to speak even faster, so any question, please don't hesitate to stop me. So as I said, inflammation is a physiological response. This was introduced by Mechnikov, and we need inflammation response, but we need to make sure that it will resolve in time. We know now from many studies in the brain and outside the brain that this myeloid cells that for years has received bad reputation as many phenotypes. They can produce neurotrophic factor, they can produce growth factor, they can be inflammatory, they can be anti-inflammatory, they can suppress tumor, they can help removing tumor. So they are very pluripotent cells, not in the sense that, but they are very diverse and they can acquire many activity. So we started this journey 20 years ago, almost 20 years ago, with a simple question. We know that the brain is irreplaceable. We can replace lung, we can replace kidney, we can replace even heart, but brain will never be able to replace. So it didn't make sense, at least to me at that time, now it sounds obvious, that a tissue which is so indispensable has given up the opportunity to be assisted by the systemic immunity, which assists every tissue repair. So that was our fundamental question, and we asked ourselves, what have we missed? Because if this is evolutionary, a mechanism to protect the individual, it doesn't make sense. So we thought that we have missed something in the interpretation. So we started in a simple model of spinal cord injury or optic nerve injury. We do know that every individual who is, God forbid, a victim of spinal cord injury or head trauma or any CNS injury, it will lead to paralysis and very limited repair. Over the years, several mechanisms have been introduced as an attempt to explain why there is no recovery in the CNS. It was discovered by the group in Germany, in Switzerland now, Martin Schwab, that myelin-associated protein are inhibitory for axonal growth. It was suggested by the group in the States and also the group in UK that around the lesion site, a scar tissue is being formed, which is made mainly by chondroitin sulfate proteoglycan, which impede growth. And there was some explanation that maybe the neuron has intrinsic inability to regenerate in the CNS. In all of the cases, there was also observation of local inflammation. Needless to say that overcoming the scar tissue was not sufficient to cause repair. Overcoming the myelin-associated protein was not sufficient to induce repair. And my team was focusing on trying to understand the local inflammation for a simple reason. When it was discovered, there was a big clinical trial in the States, which was with the blessing of the NIH, that ended up with the idea that all victims of spinal cord injury should be treated immediately with steroid for the first few hours in order to improve repair. And it was compulsive in all Western countries. Over the years, it turned out that it was not beneficial and increased morbidity of the patient. So after spending many years on this topic, we came into the idea that both inflammation and scar tissue are entering phases in response to any injury. If you follow a scratch in the skin, you will see that there is a scar, which is an interim response. You will see immune cells, which are interim response. So what is happening in the CNS, what we observe and what we hypothesized, that the scar is needed, inflammation is needed, but they are not resolved in time. So what we did and others, we follow, as you probably know, if you have an injury in peripheral nerve, there is a recovery. Only in the CNS, there is no recovery. So we compared peripheral and central, we and others, it's not only my team, and we observed that in recovery from peripheral nerve, following peripheral nerve injury, there is a local inflammation, immune cells are getting into the site of injury, and they are needed for repair. If you prevent entry of macrophages to the site of injury, there will be no repair. So inflammation is needed in peripheral nerve. The interesting part is for you as a student, we and a group in UK, Simon Gordon and Hugh Perry, found exactly the same observation, that in the CNS there is less inflammation than in the peripheral nerve. Their interpretation was that this is for the benefit of the CNS. Our interpretation was that this is against the CNS and we have to bring more macrophages. So both observations were identical, the interpretation was opposite. And that's what stimulated our work, and retrospectively it turned out that we need macrophages for CNS repair and microglia are insufficient. Is it clear so far? No question. Because it's too fast or it's too clear? No, it's clear? Okay, so I'll continue. So the first, and here I'll give you only one slide because otherwise we'll have to spend several days. So our first assumption was that what is missing in the CNS are in more macrophages, and what are missing macrophages that will resolve the inflammation. So what we did, we activated ex vivo monocytes to become anti-inflammatory. We injected them around the lesion site, and we observed better repair. So this was the first time that anyone was daring to introduce macrophages to the CNS. We found over time that the site where we put the macrophages, the timing and their number was very critical. But at that time, we didn't understand why there is no more macrophages that enter spontaneously, because their number in the blood is not limited. How do they enter to the CNS? But this was observation that made at that time. Since we saw it, we know that in wound healing, there are phases of recovery. There are first macrophages that are cleaning the site of injury, macrophages that resolve the site of injury and the inflammation, and adaptive immunity as well. So we wanted to see whether we need all the phases also in the CNS. To make a long story short, right after we found that the macrophages are needed for repair, we found that also T cells are needed for repair. And specifically what we found, that these are T cells that recognize CNS antigen. In other words, autoimmune T cell. Needless to say that it came as a big surprise that we are saying that T cells are needed and autoimmune T cell, two forbidden phenomena. And we coined the idea of protective autoimmunity because the phenomena of protective immunity is known. But protective autoimmunity was kind of a new phenomenon. I told you at the beginning that at the early stage when we introduced this idea, it was completely unacceptable to the extent that I was cited only in the negative way at the first time, the first few years, and now it's cited in hundreds time. Anyhow, at that stage, we stopped and started to think. What is the role of the macrophages? What is the role of the T cell? What are the relationship between the T cell and macrophages and the microglia? So in order to understand this, we design mice where you can label genetically only the myeloid cells that comes from the blood. So we now have macrophages that are monocytes from the bone marrow that are green, and the microglia are not green because when we did this work, there were no way to distinguish between the resident microglia and the infiltrating macrophages. And any textbook or any paper that you will read from that time or up until 2 or 3 years ago, inflammation in the brain will tell you microglia / macrophages because they were indistinguishable. So we use a chimeric mice where the microglia are not green, the monocytes are green, and we found that after injury, the monocytes from the blood enter spontaneously into the lesion site, and they are circumventing the lesion site. They are not entering inside. That's what we thought at that time. So this is how it looked like. And we also, this is our model. And we protect the head in order to make sure that we don't kill any microglia inside the brain. But because the turnover of the microglia is long, but we may kill them. So you can see that the chimeric is not 100% because this part is protected. This is the device that we developed. And from this experiment, we learned 2 issues. First, that monocytes are coming from the blood after injury. And secondly, in the healthy brain, you don't see a single green cells. That supported our notion that the microglia are self-renewal and macrophages are not replacing the microglia. So this is how it looks like. The brain, healthy brain, no monocytes. Following injury, there are monocytes. Then we devised another pair of mice where the monocytes that coming from the blood express diphtheria toxin receptor. So we can kill them selectively without killing the microglia. Is it clear? So we found that the spontaneous recovery is worse when we're depleting the monocytes from the blood, which told us that the monocytes are needed for repair. And then what we found, the question was, what is indeed their role? And what we did is the following. So I'll just summarize one work that took a graduate student 4 years to do it. So this is recovery from spinal cord injury. This was the spontaneous recovery. This doesn't work anymore. So this is the spontaneous recovery. I can manage without. That's okay. So this is the spontaneous recovery. When we depleted the monocytes, no recovery at all. When we reconstitute with monocytes that don't express diphtheria toxin receptor, a full recovery, because you don't deplete them because they don't express diphtheria toxin receptor. When we reconstitute with monocytes that we took from IL-10 deficient mice, there was no recovery. And since IL-10 is anti-inflammatory, what this taught us that macrophages are needed for repair. They are resolving the local inflammation, which is dependent on expression of IL-10. So this was this work. In addition, when we boosted on the left side, when we boosted the level of T cell that recognize a myelin antigen or CNS antigen, at any given time, we saw more recruitment of monocytes, which gave us, again, I don't want to get into details, but what we learned from 4 years of doing this work, that the monocytes are contributing to the resolution of the inflammation, and the T cell facilitate recruitment of monocytes. And again, we didn't know how T cell facilitate recruitment of monocytes, and more critically, we didn't know how monocytes can get into the CNS without breaching the blood-brain barrier. Is it clear? So from this set of study, we learned monocytes are spontaneously recruited to injured CNS. They are needed for resolving the inflammation, and T cell facilitate recruitment of monocytes. We subsequently did a similar work in the optic nerve and got the same phenomenon. At that time, I had 2 graduate students that kept coming every night to the lab with me and kept asking me why there is no spontaneous recovery. If we need T cell and monocytes and their number in the blood is not limited, why don't we get better recovery? And I told them we have to understand how they get there. But one of them, who is now independent, the 2 of them, one is independent professor in Virginia and one is a tenure track at the Weizmann Institute, told me, let's think maybe the adaptive immunity, the T cell, have more fundamental role in the CNS, which is not sufficient in injury because in injury requires more plasticity than normal plasticity. We liked the idea, and we started to think, how can we measure plasticity of the brain if the immune cells is needed for plasticity? And what we did is the following. I'll get into it later on. So what we did is the following. We said, okay, maybe it's needed for neurogenesis, because neurogenesis is formation of new neurons in the brain, and it's one of manifestation of brain plasticity. And we knew at that time that neurogenesis is associated with coping with mental stress. We knew that it's associated with learning and memory. So we said, okay, let's see if adaptive immunity affects formation of new neurons. Just to those of you who are not familiar with neurogenesis, we have different type of neurogenesis. In the brain, we call them more progenitor cells rather than neural stem cells. They have all the features of neural progenitor cells or stem cells. They are asymmetric precursor, they have the phase decision, they have the dendritic extension, they are facilitating synaptogenesis, and we know that every single stem cells in the brain can give rise to the 3 lineages, to astrocytes, oligodendrocytes, and neuron. So that fits stem cells or neural progenitor cells. We knew when we started this work from the work of Rusty Gage in the States and many others. As a matter of fact, before Rusty Gage, there was a scientist whose name is Altman in '62. He had a paper in Science showing for the first time that there is formation of new neurons in the brain, but he was totally ignored. And this was taken over by a beautiful work which was done by Rusty Gage, but Altman was the first one to discover. And we have a tendency to forget. So we know we knew when we started to think about the involvement of immune system in neurogenesis, that neurogenesis is affected by enriched environment. When we do sport activity, mental activity, increased formation of new neurons. And when we are exposed to stress, mental stress, developmental stress, there is a reduction in neurogenesis. So we thought that this is a good target to check if there are formation of neurons affected by the brain, by the immune system. And also, another reason for it is because we know that immune cells are affecting, in wound healing, affecting neural progenitor cells. So we thought this is a good idea. And so what we did to this end, we put the animal in enriched environment, the same cage that was designed by Rusty Gage and Karl Kemperman when he was a postdoc with him. Now he's in Germany. Enriched environment, you allow the animal to be in a good social life, doing a lot of exercises, which is reminiscent of our sport activity. We only put female because the male eat each other. But women are passive aggression, I always say it, so. Anyhow, we put many animals together. We observed the same phenomena they reported, increased neurogenesis. You measure neurogenesis by injecting BRDU, which measures proliferating cells, and you label for double cortin, which is a marker for newly formed neurons. Increased neurogenesis. However, when we put immune-compromised animal, SCID mice, severe combined immune deficient mice, or any type of deficiency in adaptive immunity, no increased neurogenesis, which kind of suggested to us that between our sport activity and neurogenesis is the immune system. And you can see it nicely, even without sport activity, healthy brain and the SCID mice, healthy brain, but compromised immune system, significant reduction in neurogenesis. We took another pair of SCID mice and we found the same phenomena. We also did it, we took different type of immune genetic engineered mice that don't have at all T cell that recognize self. Normal mice, reduction of neurogenesis. Mice that have overexpression to T cell that recognize brain antigen, increased neurogenesis. So overall, to our surprise, this was the first time that immune system was suggested to be associated with neurogenesis. And it's kind of anecdote. When we published this paper, it was after 2 papers that were published, one by a group in Berkeley and one by a group in Sweden, in Lund. And they were almost at the same time, one in Science and one in PNS, that inflammation impairs neurogenesis. So, yes?
When you transfer T cells, recognized over albumin, it goes down.
Of course, because it's like immune deficiency.
I guess you transfer the...
No, it's not transfer. It's a genetically engineered animal. They don't have T cell that recognize self. So it's like SCID mice.
Have you tried to transfer?
No, we did in wild type mice, we transfer, yeah. In this paper, we also transfer T cell, not to this, to T ova, but to SCID mice, and we restore neurogenesis.
But if you transfer T ova, you shouldn't rescue.
You're right, but we didn't do this experiment. Yeah. So we are also found that it's associated with increased production of BDNF. And when we tested cognition in these mice, we found the same phenomena. When you took mice, we are using a maze which allow testing ability of the animal to learn, navigation, and to remember. So you put them in a water pool of water. There is a platform in one part of the water pool. There are pictures around the pool. So at the first time that you put the animal inside, they are swimming randomly till they touch the platform. After several days, when you put them, they swim directly to the platform, not because they change the speed, but because they recognize the location of the platform relative to the picture. In immune-compromised animal, they never learn. Here we did what you asked. We took SCID mice and transferred T cell, and we did the opposite. We took wild-type mice and replaced them with bone marrow from SCID mice. So in both directions it worked. That cognition is going down. Although the brain remained the same, impairment of immune system impaired cognition. And this is in connection with your previous talk. There was an article lately which we wrote on it, News and News, showing that between the immune system and the brain, there is the microbiome. But this is just because of your previous talk. An anecdote to the women in the audience, the female in the audience. When we did this work, one of my graduate students was pregnant, and she told me, wow, I have a loss of memory. I said, okay, you're not the only one. All of us went through this stage. And she said, does it connect to what you're saying? I said, of course. Because when we are pregnant, the adaptive immunity is going down to avoid rejection of the fetus, because the fetus is 50% comes from the other part of you. So adaptive immunity is going down, and this is why cognition is going down. So we tested it, and we took pregnant mice, and we tested neurogenesis during pregnancy, and it went down completely. And after labor, it's going up. So it's a test of life. Anyhow, also, before we are entering to the second part of my talk, we found that immune-compromised mice not only suffering from cognitive loss and not only suffering from reduction in neurogenesis, they are less able to cope with mental stress, and exactly the same phenomena. And the model we adapted from a group in Ben-Gurion University, but the same model was developed also at the Rockefeller by Bruce McEwen. So if you expose mice to smell of cat, I've never seen the cat, they are stagnated, and this was done by several groups. We didn't invent the model. And the mice are stagnated, and you can measure symptoms of post-traumatic stress disorder by measuring the time that they spend in open versus a dark arm, or how much they are irritated by noise. And SCID or nude mice, SCID suffer from T cell and B cell deficiency, nude mice only T cell. We found that they are developing symptoms of post-traumatic stress disorder, but I don't have time to get into the immunology of that. In addition, so what I've shown you so far, that adaptive immunity, specifically T cell, facilitate recruitment of monocytes in a mechanism that we don't know how. I showed you that the immune system support cognitive ability and neurogenesis in a mechanism that we didn't know at the time how and from where, because they are not in the brain. And I'll show you that they are affecting neurogenesis. So in order to further understand neurogenesis, we decided to see if the stem cells in the brain, the neural stem cells, express any activity which is reminiscent of immune cells. And we focus on toll-like receptor, and this was a graduate student that was in my lab when we did this work, and then did a postdoc in Stanford, and now she's back in Israel in the Technion, and she monitored whether stem cells are expressing, neural stem cells in the brain are expressing toll-like receptor. And I just want to show you one slide. So she found that neural stem cells in the brain express all toll-like receptors that immune cells express, and they are very much sensing the environment in a similar way to the immune system. So overall, from what we learned from this journey, that the brain is not escape from dialogue with the immune system. And the question was where all it happened and how do they enter. And it summarized all the protective autoimmunity phenomena that we found over the years. And now it's repeated by many, many other groups. The major, really one of the star in the group is now former graduate student from my lab who is now in Virginia. And he also observed that there is a lymphatic drainage in the CNS. So overall, what we found that immune cells support, circulating immune cells support healthy brain maintenance, cognition, neurogenesis, and helping repair. So left with all of these questions, we started to decide first to see how do they get into the brain. And again, this took us 5 years, but finally we found that even if you inflict injury in the spinal cord, the immune cells that contribute to the resolution of inflammation don't come from the site of injury. They are coming from the remote choroid plexus epithelium. So less than 24 hours after the injury, we see that the barrier that I started with is activated. You can see this is the barrier when there is no injury. Here, the remote barrier, it doesn't suffer from the injury, is activated and all of a sudden express leukocyte trafficking molecule. So we characterize the leukocyte trafficking molecule. I will not get into it. We found that the monocytes are really entering through the remote gateway. And when we block, we characterize numerous trafficking molecules expressed by this barrier, which are remote from the site of injury. Here at the site of injury, these are the 2 ventricles in the brain, and the third and the fourth ventricle. And we found that the site of injury here and the cells are crawling through the central canal and getting into the site of the injury. So, and when we block these trafficking molecules that express at the choroid plexus, there was no repair. So our suggestion was that at least some of the immune cells that are entering to the CNS after injury are entering through the remote choroid plexus epithelium. Retrospectively, evolutionary and philosophically, it makes sense because it's a kind of a safety mechanism that by the time that they reach the lesion site, they are educated not to affect repair, to become pro-inflammatory. So this is how it looks like, that the choroid plexus is permissive for entering of immune cells, and the injury activate this gateway. So this is the structure. Sure. Sure.
Just a matter of definition, the choroid plexus is the entire epithelium lining of the entirety of the ventricle.
Exactly.
It's not a particular region of the ventricle.
It's entire. Yeah. And then you have the meninges, but it's the choroid plexus epithelium. The meninges is endothelial barrier. Yeah. Sure. Any other question? So now we are getting into the interesting part. Other questions?
Recruit memory. Recruit memory cells more than.
Oh yeah,
cells.
I'm getting into it. Exactly. Good question. So this is the structure of the choroid plexus. So these are the villi of the epithelium that facing the CSF. This is the stroma, and this is the fenestrated blood vessels. So in the stroma, it was already reported before we enter into this area that there are antigen-presenting cells, but no evidence of T cell there or anything like that. So I told my people, let's start to understand this tissue. It's so interesting. Let's start to see what is this tissue. So it looks like that. And the ZO1 is for the connection, and the CD3 are the T cell. And what we found, and the CD31 are the blood vessels. And what we found that even if we intensively perfuse the animal, we found this T cell outside the blood vessel in the stroma. So we isolated the T cell, and there are about 250 T cells in each choroid plexus. 250, so not many of them, but we were very excited. This is how it looks like. And now I'm getting into the next second lecture. So if there are any questions at this stage, everything is clear. Yes?
Are the scars in the central nervous system caused by both trauma and disease?
Yeah. It's a very good question. I skipped it because of interest of time, and I thought that I'm overloading you. It's a very good question. The glial scar is formed in the CNS under all pathological conditions. We were amazed to see how massive is the glial scar in Alzheimer's disease. I'll get into it later on. And what we found, this was in PLOS Medicine 2009, what we found that the same cells that resolve the inflammation are also producing metalloproteases that contribute to the scar resolution. So if you bring more macrophages, they resolve the inflammation and resolve the scar. However, in order to acquire a phenotype that will allow them to resolve the scar, they have to encounter the scar. So the scar is using educate them and is resolved by them. So it's like a loop. Yes?
You said that in the spinal cord you had the healthier results, but did you manage to bring back the spinal cord? I probably missed it.
In the spinal, what did I say?
Did you manage to bring back the spinal cord?
In spinal cord, even if the best neurosurgeon in the world will sit down and suture the two ends, it will never recover.
Okay,
You have to grow back all the way.
So when you say recovery, what did you mean?
Okay, so recovery in the CNS means three. One is full regeneration, which is hardly happening. There is a lot of collateral loss following spinal cord injury or any CNS injury because the primary damage cause chaos in the environment, including inflammation, which cause further loss beyond the primary loss. And when you resolve the inflammation, you can rescue neurons that have not been damaged, and you allow some collateral regeneration. I didn't want to get into it, but yes.
So you mentioned there are 250 cells.
I'm just now getting into this.
Oh.
Exactly to
So
this.
probably means there's no local expansion.
This is a good point.
Yeah.
I really don't.
Sorry, you mentioned you had a mice that you have T cells recognizing the autoimmunity or something.
I'm getting into it. Okay. So because we observe T cell here, and because what you just said, that we observe early on that what we need are autoimmune T cell, we decided to isolate this T cell. So we isolate them, and by flow cytometry, we found that almost 97% of the T cell are effector memory T cell, which means they are not naive T cell, but educated T cell. And then the question was whether these are CNS specific or not. That was your question, right? Okay. So 97% of the cells are memory T cell. They are engaged because they are memory T cell. They need to be engaged with antigen presenting cells, otherwise they will not survive because memory T cells are consuming IL-2 that they are producing. It's kind of autocrine regulation. So we found that they are indeed engaged with dialogue with antigen-presenting cells. You can see MHC class 2 and CD3. These are antigen-presenting cells. These are CD3. And we found that they locally produce interferon gamma, IL-4, and IL-10. Now the question was, what is their specificity? Now, those of you who are not familiar with immunology, the T cell recognize a complex antigen. So it's not like B cell that the receptor is immunoglobulin. So you don't know what they recognize unless you know with what you vaccinate. So when you have a pool of T cell, you don't know what they recognize. And we envision that maybe they are recognizing CNS antigens. So to do that, we took spinal cord, which contain all CNS antigen. It's like a mesh. And we immunized naive animal. We isolated the spleen, collected all the CD4 positive T cell from the spleen, and did T cell receptor library. Now we took naive, and this is back to your question, we now took the T cell from naive animal and create their T cell receptor library. And by comparing this, we could tell whether the T cell in the choroid are enriched with CNS specific T cell. Is it clear? And we found that 70% of the T cell in the choroid plexus are recognizing CNS antigen. I don't know the identity, but they recognize it.
One more thing, there's only one thing for me anyway that's not clear.
Only one thing.
Only one thing that I know about.
Okay.
The T cells you're referring to anatomically within the epithelium, where are they located? Are they exposed to the CSF or are they not exposed to the CSF?
That's a great question. Probably not exposed to the CSF.
At least on the average.
On the average, but I'll show you.
Right?
There are many questions that I don't know.
Yeah,
Okay, so but they are sitting there, they are producing cytokine, I'll show you in a few minutes. The epithelium, the side facing the CSF, is sensing what's coming from the CNS, and from the other side, they are sensing cytokine. So the integration between the two activate the choroid plexus.
So there's no way is it continuing to pick up antigens coming.
They are not picking up antigen. I'm almost positive about it. I didn't check, but I'm almost positive. I think that it's one-way ticket. They are going out, but they are not going. From the choroid plexus, for sure, they are not getting out.
Good, thank you.
But the specificity is this, the specificity. And now, back to your question. The question was what they sense. So we made a monolayer culture of choroid plexus epithelium, and we let them really reach tight junctions. And what we found, we decided to expose them to the cytokine that can come from the T cell or the cytokine that come from the CSF. Now, the cytokine that come from the CNS, we envision that this is what comes after injury, because that's when the choroid plexus is activated. And what is common in injury is IL-6, IL-1 beta, and TNF-alpha. So we cultured the cells, and what we did, so we thought, okay, if there is a pathology, SOS signaling will be IL-6, IL-1 beta, TNF-alpha, and the T cell will produce either interferon gamma or IL-4 or IL-10. These are the major T cell-derived cytokines that we found. And we measure expression of trafficking molecule. I'm showing here only two. And what we found, to our major surprise and a lot of satisfaction, that TNF-alpha can only activate the gateway to very slim response. Interferon gamma out of the T cell is the only cytokine that activates the gateway. But there is a huge synergy between TNF-alpha and interferon gamma, which means that when there is a pathology, TNF-alpha from inside activate the gateway, and the interferon gamma from the other side activate, and together they are allowed trafficking. And what we found that the TNF-alpha upregulate interferon gamma receptor on the epithelium. And that's how they work in synergy. And you can see it here. So the TNF-alpha upregulate interferon gamma receptor and also IL-1 beta. So that's the basis for the synergy. So also to find out whether TNF-alpha is sufficient to activate the gateway, we injected TNF-alpha stereotactically into the CSF. And we found activation of the gateway and trafficking of immune cells into the CSF. So all of this brought us to think that maybe the choroid plexus is the nexus between the brain and the circulation, which may have a key role in brain aging and in neurodegenerative diseases. So we decided to go to aging first because this is a wild-type animal. And we monitor level of interferon gamma in the choroid plexus with aging, and we found that between 12 and 18 months, there is a dramatic drop in interferon gamma availability in the choroid plexus. About the same time, there was a paper from Tony Weiss-Curry in Stanford that aging of mice is associated with elevation of the chemokine CCL11 in the blood and in the CSF in mice and humans. So we decided to see whether CCL11 is produced by the choroid plexus and whether it's produced in aged choroid plexus. And we found that this is, again, indeed the phenomenon. Choroid plexus of young animal don't express it, old animal express it.
So have you done your experiment in our TNF-alpha or gamma knockout mice?
We did, but I don't have time to show it, and it was published. Interferon gamma knockout recover worse, less immune surveillance, all of it, but I don't have time to cover it because I want to move into the aging and Alzheimer. But you can send me an email and I'll send you all the papers. So in order to address the issue of aging, what we did to see whether the choroid plexus is the missing link in aging, we took 11 tissue from young animal and 11 tissue from old animal and did robust RNA sequencing. And we separated the choroid plexus from the brain of course, and from the circulation. And this was done in collaboration with Amit. We have graduate student between the two of us. And we found that the most surprising, I must admit, that the choroid plexus in aging show elevation in interferon beta. Interferon beta, it's antiviral response. And we didn't find it in any other part of the body in aging, and we didn't find it in young animals. So I always say that when we saw it, we blame each other that our colony is contaminated with interferon beta. So we ordered mice from several places, and it was absolutely reproducible, including the mice from Stanford, because later on we collaborated with the group in Stanford, and it's the same phenomenon. And interestingly, along with the elevation of interferon beta, there was a loss of interferon gamma, which we already knew. So this was a sanity check for us that this is right. And a couple of months before our science paper, there was another paper in Science showing that there is inverse relationship between interferon beta and interferon gamma. So we thought we have an interesting phenomena. We got human section, a post-mortem, from a UK center, and we asked them to send a post-mortem section from brain of aged people that didn't die out of any neurological disease, and we found the same signature, interferon beta signature. So we continue, and this is to your question, we checked interferon gamma knockout mice receptor or a cytokine. We found reduction in cognitive ability and reduction in neurogenesis. Now, together with Tony Weiskoury in Stanford, we decided...
Can I ask you one more
Sure.
question that underlies a whole lot of things here?
Yeah.
A little bit general. Is in this or pregnancy or what have you, the relative diminution of cognition, is it due to, on the one hand, damage that's ongoing at a certain rate that's not being repaired, or is it due to a positive increment that does not involve damage, neurogenesis, above and beyond the physiological?
That's a good point. The interferon gamma, what we see that if you take healthy animal, interferon gamma knockout, in 3 months, no effect on cognition. 6 months, 3 months, even better cognition. 6 months, no effect on cognition. 9 months is premature aging of the brain, which means that interferon gamma is part of the surveillance of normal healthy. So it's superior.
So if you could, would you recommend interferon gamma injections in everybody? It's really what I'm coming down to. Is it damage that's going on that's differential from one individual to another, and then you're replacing damaged neurons? Or is it that you're helping form new pathways separate from any
I think that I will show later on some signature on microglia. The short answer that I really don't know.
Okay, that's fair.
But we have...
It's interesting either way.
It's interesting. But we have an indirect indication showing that because the lack of interferon gamma is not affecting 3 or 6 months, it means that at 9 months onward, which is the equivalent of middle age in human, you have increased susceptibility, which means that interferon gamma is part of allow the trafficking immune cells for surveillance and maybe repairing minor damage rather than just a mechanism that is superior.
That's what I would have guessed.
Yeah, this is our guess, and now we are checking what makes neurons or microglia at middle age more susceptible than young.
Okay.
And we have some data, but very preliminary.
Thank you.
Sure. So regarding the interferon beta, what we did with Tony Weiskoury, we wanted to see whether the interferon beta, and this is back to your question, whether the interferon beta going up is induced by the brain, something is wrong in the brain, or in the periphery. So we did parabiosis mice. So we connected young mice to aged mice. The connection is through the skin, and then they are sharing the blood, but they are not sharing the brain. So it's sharing only the blood. In control, we have age connected to age and young connected to young. And what we found that the aged microglia, the aged mice maintain the interferon beta signaling, which means that interferon beta signaling is induced by something which going in the brain. And what we did to address this, we cultured choroid plexus from young animal and exposed them either to CSF of young or CSF of old. And we found that the CSF of old mice induced interferon beta. And we could mimic it by TNF-alpha or IL-1 beta. So in other words, what we believe that the immune cells help from the circulation, help restoring maintenance of the CNS. To see whether the interferon beta from the choroid plexus is responsible for, the interferon beta is responsible for the loss of cognition. What we did, we injected antibody directed against interferon beta receptor because the choroid is producing interferon beta and responding to, because it's an autocrine phenomenon. So we do two things. One, we neutralize interferon beta receptor on the choroid plexus, and also which neutralize also interferon beta receptor on microglia in the brain or any other cells in the brain, because we injected antibody directed to the receptor. So you can neutralize the choroid and look, is it clear? Or I'm getting too much? Clear? Okay, so we injected the antibodies and we restore the choroid plexus epithelium. And what we also restore cognitive loss. So all aspects of choroid plexus were restored by injection of the antibody against interferon beta receptor. And also we restore cognition. Again, we checked, since they are aged mice, we are using ARENA, or which is called novel object location. We changed the location. We took a big cohort of mice, first test their cognitive ability, and we took out of them only the 70% of the mice that lost cognition, and then we separate them. Half of the mice received the IgG, and half of them the anti-interferon, and we restore cognition. We wanted to see whether we also restore microglia. So we did RNA-seq of the microglia that we isolated from the brain of aged animal and young animal. And first what we saw that the signature of aged mice of the microglia is mainly of interferon, type 1 interferon. So the same interferon that produced by the choroid plexus is the signature of aged microglia. And what we found that when we reversed when we injected the anti-interferon, many of the genes that are known in microglia that are impairing cognition, such as complement, such as a beta-2M, are restored in the microglia of aged mice that when we neutralize the interferon beta receptor. So I'll skip this and go to the last part. Okay, so I'll skip this. Maybe if we have time, we'll return into it. So based on all of this study, what I'm demonstrated to you, the choroid plexus is the interface between the brain and the circulation, and the choroid plexus very much affect the fate of the brain. And in aging, there is shut off of the choroid plexus because of elevation of interferon beta. So we thought this is a great opportunity for us to challenge this part in Alzheimer's disease. Now in Alzheimer's, so the last part will be devoted to Alzheimer's. In Alzheimer's, we know that there are many features that are considered as hallmark of Alzheimer's. There is accumulation of misfolded protein known as amyloid plaque. There is a local inflammation. There is also tangles in the brain, in the neuron, because of tau pathology, which is known as that microtubule lost their structure. And there are some evidence that there are a problem with neurotransmitters, such that the only treatment, symptomatic treatment that currently exists to Alzheimer's is acetylcholine esterase inhibitor. So as I said, the many attempts were done to suppress the neuroinflammation by systemic anti-inflammatory, and it didn't work. And more importantly, numerous attempts for many years were done to remove the plaque there was recent clinical trial which was very productive in eliminating the scar, the plaque, but it was not sufficient to modify cognition. And the question was, what have we missed? They are not contributing cognitive impairment. And according to our understanding, by the time that you have plaque, there are numerous other collateral factors that contribute to disease escalation, and therefore removing of the plaque is insufficient. So based on our understanding and numerous work, not only of us, have shown that recruitment of macrophages is needed to remove the plaques. And this is just to show you how plaque is engulfed by macrophages. So we thought if we need removal of the plaque, we need macrophages. And I demonstrated to you an hour ago that we need to activate the gateway for entry of macrophages. We took a mouse model of Alzheimer's, which has 5 human mutations, 3 of A beta and 2 presenilin. And it mimics very nicely the Alzheimer's disease in patients, except that it's developed early in life. There is reduction of neuron, loss of neuron, loss of synaptic plasticity, aggregation of beta amyloid. So we took this mouse and we found that at any given time that we tested, the trafficking molecule expressed by the choroid plexus are lower than even wild type. The broken line is age-matched wild type. So loss of expression of trafficking molecules. When we saw this, we got human section and found the same phenomena, loss of trafficking molecule. We found that in the mouse model that we used, the loss of trafficking molecules was associated with loss of interferon gamma at the choroid plexus. Alzheimer's, loss of trafficking molecule, correlation with interferon gamma, which was very nice because we demonstrated we need interferon gamma. So immediately there was another report in the literature that found exactly our phenomena using a different mouse strain of Alzheimer's, loss of interferon gamma and loss of trafficking molecules. So how can we circumvent loss of interferon gamma? So interferon gamma is a product of effector T cell, and in the body they are suppressed by regulatory T cell. They may be suppressed also by checkpoint, inhibitory immune checkpoint. And those of you who are not familiar, effector memory are kept under constraint by pathways that are inhibiting them to avoid overshooting of immune response. They are known mainly in connection with cancer because the most known therapy now, immunotherapy for cancer, is anti-checkpoint antibody or inhibitory antibody directed to inhibitor immune checkpoint to unleash the immune system. So when we saw that the choroid plexus is suppressed, deficiency of interferon gamma, we decided to go in the 2 directions. Remove regulatory T cell or unleash the immune system by using antibody directed to inhibitory checkpoint. So the first thing, we got mice from Rodensky in the States, and we bred the Alzheimer mice with mice that allow us to deplete selectively only suppressor cells because they express diphtheria toxin receptor under the transcription factor 4p3, which is the transcription factor which regulates regulatory T cell. We depleted the regulatory T cell. In this mice, you can deplete only once because the regulatory T cell, the remnant regulatory T cell are proliferating and they don't express anymore the diphtheria toxin receptor. So we did this. We found that trafficking molecules went up as a result of the depletion of the regulatory T cell. We see it by mRNA and we see it by immunohistochemistry. The gliosis, back to the question early on, if there is gliosis in Alzheimer's, here you can see gliosis. The lady over there, she's sitting with her iPhone, but this is the gliosis. Okay. So it's boring, maybe because of that you're sitting with okay. It's too much. I'm finishing in a few minutes. So I think that 1.5 hours is a long conversation, a long discussion, a long talk, right? Okay. So we have here, I've never spoken so much. So we have here gliosis, which is going down. And we have dramatic reduction in plaques. So the A-beta plaques, since they are human mutation, you can stand them with antibody directed to human A-beta. So here in the cortex, you have many spots. These are the plaques. And this is in the dentate gyrus. And the treated animal, the plaques are almost gone. When we tested cognition, this was a big surprise because this is more water maze. So the wild-type animal, the black, are learning and they remember. The Alzheimer mice remain stupid. They didn't learn. And it's so sad to say stupid. So they didn't remember. And when you take out the platform and you measure the time that they are insisted that they remember where the platform was, they didn't remember. Whereas the animal that were depleted, the suppressor cells, learn as good as the wild-type animal. So we treated the animal at the stage where the memory was completely lost and they regain it, which was surprising. We'll get into it in a few minutes. And we saw in the brain a month after the treatment, we see immune cells are circumventing the plaques, both suppressor cells and are also myeloid cells. Based on this, we move further to check checkpoint. And the inhibitory checkpoint that we thought that are relevant to us are the PD-1, which is programmed S1. So they are expressed on effector T cell of the family of CD4 and CD8. CD8 is relevant to cancer, CD4 is relevant to our story, and they mainly suppress autoimmune cells, so that's why we were very excited to think about it. Now, the ligand can be expressed on epithelial cells, which for us is our target of the T cell. They can be expressed on regulatory T cell or antigen-presenting cells. So all of these are present in the choroid plexus epithelium. We have the epithelial cells, we have the regulatory T cell, and antigen-presenting cells. So we decided to inject antibody directed either to PD-1, the receptor, or to ligand, and see whether they will release the immune system in order to help in Alzheimer's. In the meantime, we checked to see whether the PD-L1, the ligand, is expressed by the choroid plexus, and we saw that it expressed, and it's going up with aging. So the animal received antibody directed against the inhibitory immune checkpoint, and we were hoping to see whether interferon gamma is going up. And we found that both by RNA-seq of the choroid plexus and by measuring CD4 positive T cell that express interferon gamma, that the choroid plexus was activated. We isolated the microglia separately from the macrophages, and we saw that indeed we recruited double the number of macrophages to the lesion site. We found that they express different activity. This is just to show you a myeloid cells that hug a plaque. So it's so nice that I'm always excited to see it. So microglia can hug the plaques at the early stage, and then they are losing the activity as the macrophages are needed. We checked to see whether the recruitment of macrophages was dependent on interferon gamma, unleashing of interferon gamma. So we repeat the experiment and injected antibody against interferon gamma before the anti-PD-1, and we shut off completely the recruitment and activation of the gateway. We checked cognition of this animal, and we checked by radial arm water maze because this is what recommended for Alzheimer's. So it's a maze, again, water pool with 6 arms. In one arm, there is a platform, and you will measure memory by measuring the number of errors that the animal made before they enter to the right arm. And you can see the black show you the memory of wild-type animal. The green show you the memory of those that received the anti-PD-1. The red are the Alzheimer, non-treated, and the gray are the placebo. We measure the plaques, and you can see these are animals that received the placebo. Many plaques, they will receive 2 injections of the placebo in a month's interval, so it's 2 months after we started the experiment. Many plaques, and the green are the scar. Many plaques in the cortex and in the dentate gyrus, and the treated one almost gone. We've done hundreds of animals. maybe 200 since we did it because we were so surprised with the results. Together with a group in Tel Aviv University, we checked other type of Alzheimer mice, which are double transgenic, and we found the same phenomena. We wanted to see if we start the treatment early, if we prevent loss. So you can see that we started early because we measured cognition the first time at 5 months, so the animal still have some memory capacity. If you see at 6 months, these animals are worse, whereas the treated ones still maintain very nice cognitive ability. We wanted to, because we knew from the literature that removal of A-beta is not sufficient, and because the treatment is not directed to A-beta, we wanted to see what other processes in the brain were beneficial with the treatment. So we measure neurogenesis. This is neurogenesis in wild-type animal. This is neurogenesis in the Alzheimer, treated with the placebo, and this is treated with the anti-PD-1. We measure some aspect of synaptic plasticity, and the most dramatic effect was rescue of neurons. So, of course, you cannot revive dead neurons, but we rescue some neurons. So this is the wild type, this is the Alzheimer, and this is the treated one. And since we did this work, we published this work, we repeated to see whether the antibodies are directed against the ligand rather than the receptor will do the same effect, and we found the same effect. And also because the treatment is not directed to the disease, we took another mouse strain, which is not A-beta at all, the tau pathology, which was developed here at the Hebrew University, and we found that it's as effective. So to summarize this part, and then I'll give you overall picture. So this part, we suggest that unleashing the immune system by immune checkpoint blockade can facilitate recruitment of immune cells to the brain. The antibody can work either in the lymphoid organ or in the interface of the choroid plexus, the storm between the brain and the circulation, which allow activation of the gateway and recruitment of immune cells. Now, the immune cells are not directed directly to any of the pathology, but they can facilitate removal of plaque. We have data to show that they change the milieu from less pro-inflammatory. They can rescue neuron and can rescue synapses and support neurogenesis. So if I started by saying that we absolutely in agreement with the community that there is inflammation which need resolution, the only difference that over 20 years we discover that there is a permissive gate between the brain and the circulation that needs to be activated to allow immune resolving cells to get in. And what we discovered that the immune system is the key to this gate to allow entry of immune cells. So this is I love this because when people ask me what's next, I said it's only the beginning. So this is a summary of what I've told you. So the immune system is now the best what to do. We just need to harness it at the right time. And although I gave credit to the student, I didn't give any credit to the supporting agency. Without them, there is no way to do this work. And again, of course, the student. And I'll be more than happy to answer more questions. And I hope I didn't bore you.